So we had a very exciting session actually, which I had the pleasure to co-chair with Dr. Andreas Trump, on clonal dynamic and leukemic stem cells, with a fantastic lineup of speakers, which presented very exciting data. So the first talk actually was from Dr. Ravi Majeti, and presenting data on epigenetic regulation from diagnostic to relapse samples. And while actually 40% of AML won't have any de novo acquired mutations between diagnosis and relapse, they will have an important change in epigenetic regulation. Second talk was from Dr. Liran Shlush. Very nice talk on creating an atlas from single-cell RNA-seq data from PBMC, from healthy donors, with a number which is significant, so 100 patient samples, or healthy donor samples, which should prove to be very useful actually to interpret variations that we can see during hematological malignancies. Then we had a very nice talk as well from Armela Jeremias, and actually this one was on clonal dynamic using barcoding on primary patient samples and PDX models to follow actually how the clones evolve and if any therapy will impact this architecture. Then we switched for Dr. Jeffrey Tyner, and that was a very interesting one. I mean, it's a resource which is well known. I mean, from here actually, from him, which it's a very powerful tool where they incorporated molecular data, clinical data, drug response, and trying to determine new determinants of clinical outcomes. And finally, we had a talk from Claudia Langerke, and that was a really nice one too, and where we had insight into immune escape in AML and some actually potential combinatory therapy of NK cells and PARP inhibitors. So looking forward to what is going to unfold with these new findings and exciting data.